Good afternoon. Welcome to this session of 5 days National Workshop on CSIR-UGC-NET-JRF Lectureship Preparation in Life Science, organized by Botany for You. My name is Dr. DEVI CHINMAYEE and I am Assistant Professor, Department of Botany, Shree Ayyappa College for Women, Nagarcoil, Tamil Nadu. Today, I am here to explain about light harvesting complex and photosynthetic apparatus. This is the first portion of system physiology in plants of our CSIR syllabus. So starting with photosynthesis. What is photosynthesis? Photosynthesis means synthesis with the help of light. But this term is usually applied to very important vital process by which green plants synthesize organic matter in presence of light. Photosynthesis is sometimes also called as carbon assimilation and represented by this formula, that is 6 molecule of carbon dioxide and 6 molecule of water in presence of light and green pigment present in plant produce hexose sugar. But this is a traditional or old formula which is replaced by the new one, in which the correct formula is this one, 6 molecule of carbon dioxide and 12 molecule of water. So this was one of the question in previous CSIR-UGC-NET. So from that option we have to select which is the correct formula. So 6 molecule of carbon dioxide and 12 molecule of water molecule in presence of light and green pigments and plant enzymes produce glucose or hexose sugar, and as a byproduct oxygen is also produced. So in this photosynthesis, which is a metabolic pathway, carbon dioxide along with water is used to produce carbohydrate in presence of light. Carbohydrate is synthesized along with oxygen is released as the byproduct, in which light energy is converted into chemical energy and it is stored in the organic matter. And oxygen is one of the end product. Photosynthesis, as I said earlier, it's an anabolic process in which light energy is converted into chemical energy. Chemical energy is stored in the organic matter, which is carbohydrate. So this was another one question which was asked earlier in CSIR-UGC-NET. Where is actually the most amount of photosynthesis, or the maximum photosynthesis takes place in ocean. About 90% of the total photosynthesis in the world is carried out by algae grown mainly in ocean, also in fresh water. So there will be many options such as evergreen forest, such like that, but the correct answer is in ocean. 90% of the total photosynthesis takes place in ocean. So what is the significance of this photosynthesis for mankind? As we all know, that is, it maintains an equilibrium of oxygen in the atmosphere. It also provides food. It also provides reserve energy, fuel, all the things we get is from the plant product, by plant produced by photosynthesis. So photosynthetic apparatus, as we all know, photosynthetic apparatus is chloroplast. We will go through a video for the structure of chloroplast. Chloroplast in green plant constitute the photosynthesis. The size of chloroplast is generally 4 to 5 micrometer. So size varies depending upon the species. General size is 4 to 5 nanometer. Then the plant growing in shade contain larger chloroplast in the cell as comparing to the plant which is growing in intense shade. Okay, so the size of the chloroplast may also varies depending upon the species. It is plate shaped in chlorella, and it is cup shaped in Chlamydomonas. In the case of Spirogyra, they are ribbon shaped and spirally coiled. In the Cycnema, it is star shaped, and it is generally spherical or ovoidal shape in higher plants. Chloroplast, like other organelles seen in the cytoplasm, they are a double membranous structure. That is, it is having an inner membrane and outer membrane, which constitute a disc-shaped chloroplast envelope. Outer membrane, which is permeable to almost all molecule and allow the entry of the molecule, and inner membrane is selectively permeable, and outer membrane is freely permeable. Okay. This outer membrane is having several transmembrane proteins which transfers this sugar molecule or the molecule in and out of the chloroplast organelle. The outer and inner membrane of the chloroplast encloses a semi-gel-like fluid known as stroma, and also several different organelles, plastid, DNA, ribosomes, RNA, enzymes also present inside this stroma. And there is a structure called thylakoid is also present. Internally, the chloroplast is filled with hydrophilic matrix called stroma, in which are grana embedded. Granum has a diameter for 0.25 to 0.8 microns and consists of 5 to 25 disc-shaped grana lamellae, which is also called thylakoid, placed one above other like stack of coin. In Greek, the word thylakoid means sac. The collection of thylakoid is called thylakoid system, is seen inside the stroma of the. There are generally three spaces are seen inside the chloroplast. That is intermembrane space, that is between inner and outer membrane, and inside the inner membrane there is a fluid filled space called stroma, and also another space called thylakoid lumen is also seen in the chloroplast. This thylakoid lumen is also called loculus. Okay. Chlorophyll is found in the thylakoid and it is the site of light reaction in the photosynthesis. And inside the stroma, dark reaction takes place. Light reaction in the thylakoid membrane and dark reaction in the stroma region. The thylakoids are arranged in stacks known as grana. Each granum contains around 10 to 20 thylakoids. Some of the thylakoid of the granum are connected with the thylakoid of the other granum by somewhat thinner membrane called the stroma lamellae, or it is also called intergranal lamellae. Both thylakoid membrane and stroma lamellae are composed of lipid bilayer and protein. Chlorophyll and other photosynthetic pigments are found in the form of protein pigment complex, mainly in thylakoid membrane of the grana. Thylakoids are the site of primary photochemical reaction. So these thylakoids are the site of primary photochemical reaction. Some of the protein pigment complex are also found in stroma lamellae or intergranal lamellae. The dark reaction of the photosynthesis occur in the stroma region. Besides necessary enzyme, some ribosomes and DNA also have been found in chloroplast, which give the chloroplast a partial genetic autonomy. So coming to chloroplast genome, chloroplast genome encodes approximately 30 proteins that are involved in the photosynthesis, include component of photosystem 1 and 2 of the cytochrome BF complex and ATP synthase. In addition, one of the subunit of ribulose bisphosphate carboxylase, that is Rubisco, is encoded by chloroplast DNA. Rubisco is the critical enzyme that catalyzes the addition of carbon dioxide to ribulose 1,5 bisphosphate during the Calvin cycle. So the chloroplast genome is very important for its chloroplast genome is very important because it encodes approximately 30 proteins that are involved in photosynthesis, including components of photosystem 1 and 2 and cytochrome BF complex ATP synthase. In addition, one of the subunit of ribulose biphosphate carboxylase, that is Rubisco, is also encoded by chloroplast DNA. As we know, the chlorophyll pigments are arranged in the thylakoid membrane. This thylakoid membrane is having several granular structure called condosomes. They are arranged throughout the thylakoid membrane. These condosomes are generally of two types, photosystem 1 and photosystem 2. So, condosomes can be defined as the condosomes are responsible for capturing photons of solar electric magnetic radiation and act as the photosynthetic unit. The two type of condensates, photosystem 1 and photosystem 2. Photosystem 2 is also called PS2, which is the larger molecule seen in the thylakoid membrane. Photosystem 1 is the smaller molecule which is also seen in thylakoid membrane and also in the stroma lamellae. Okay, so photosystem 2 is only present in thylakoid membrane only, but photosystem 1 is present in both thylakoid membrane and the stroma lamellae. So these are the ultrastructure of the chloroplast with the thylakoid and the stroma. Coming to photosynthetic pigments. Photosynthetic pigments are the major component of light harvesting compound which is present inside the lumen of thylakoid. There are generally three types of photosynthetic pigments. They are chlorophylls, carotenoids and phycobillins. Chlorophyll and carotenoids are insoluble in water and phycobillins are soluble in water. They show a property of fluorescence and absorb light of different wavelengths. So these are the distribution of different types of photosynthetic pigments seen in plant kingdom. So there are different types of chlorophyll molecule, chlorophyll A, B, C, D, E, F, etc. Of this, chlorophyll A and B are present in green algae and higher plants. But this chlorophyll A is present in almost all photosynthetic higher plant and algae. It is also called universal chlorophyll pigment. Okay, so chlorophyll A directly takes part in photochemical reaction. It is the reaction center in the photochemical reaction. So it is called primary photosynthetic pigment. The other photosynthetic pigment including chlorophyll B, C, D, E, carotenoid and phycobillins are called accessory pigment because they are not directly involved in photochemical reaction and can absorb the light of different wavelength and transfer finally into chlorophyll A through electron spin resonance. So, little bit about chlorophyll F, the recently discovered chlorophyll from stromatolites. That is the layer of cyanobacteria seen in water. So it is first identified by Chen et al. In 2010 and it absorbed light of infrared region. That is, they absorb light under shaded condition also. So type of chlorophyll, as I already told earlier that chlorophyll A, which is a universal chlorophyll molecule present in most of the plant. Chlorophyll B is present in green algae and higher plants. Chlorophyll C is again classified into C1, C2, C3, etc. Found together with chlorophyll A in marine algae. Chlorophyll D in cyanobacteria and red algae, and chlorophyll F is also seen in cyanobacteria. So in almost all green plants, chlorophyll A and B are in the ratio 3:1. And the chlorophyll F absorb the light which is from the infrared region. Normally the chlorophyll molecule absorbs light from visible region of the spectra. But chlorophyll F, which is an exceptional case, absorbs light from infrared region. Coming to structure of photosynthetic pigment. And chlorophyll molecule is also called magnesium porphyrin compound. It is having four pyrrole ring and I have a tadpole-like configuration consists of porphyrin head and phytol tail. The porphyrin head is made up of four pyrrole ring and interconnected by methane group. In the center of the pyrrole ring, there is a divalent magnesium is present. The phytol tail also consists of 20 carbon long chain. So, the molecular formula of chlorophyll A and B are given here. So, chlorophyll A is having molecular formula C55H72O5N4Mg and chlorophyll B formula is C55H70O6N4Mg. It was one of the previous year question of part B in CSIR-UGC-NET. So these are the structure I was telling earlier, that is so coming to structure of chlorophyll molecule, which was first put forward by Willstater, Stroll and Fischer in 1912. So chlorophyll molecule are magnesium porphyrin compounds. This porphyrin ring consists of 4 phytol chains, and these are attached by a CH bridge. A long chain of carbon which is attached to the 4th pyrrole ring. It is an hydrocarbon tail, also called phytol chain. Okay, so this magnesium porphyrin head is hydrophilic and phytol chain is lipophilic. Hydrophilic means water loving and lipophilic means lipid loving. This hydrocarbon tail is 20 carbon length tail, which is seen attached to the 4th phytol ring of the chlorophyll pigment. So the chlorophyll is a chlorine pigment structurally similar to porphyrin pigment such as heme. In heme, the central atom is iron, and in chlorophyll, central atom is magnesium. It is structurally similar to heme. So two different types of chlorophyll, major type of chlorophyll, chlorophyll A and B. Different is that the CH, the aldehyde group is replaced by a methyl group in the 3rd carbon of the 2nd ring in chlorophyll B. In chlorophyll A, methyl group is present, and in chlorophyll B, an CHO group is present. This is the only difference between chlorophyll A and chlorophyll B, which is a structurally similar photosynthetic pigment. So these are the different chlorophyll pigment and its structure, and it is similar to chlorophyll A. The structural formula of chlorophyll C and chlorophyll D are also known. Chlorophyll C different from chlorophyll A in lacking phytol chain. Chlorophyll C lacks phytol chain. Chlorophyll D differ from chlorophyll A in that ethyl group is differing in chlorophyll D, and it is replaced by an CHO group in the 2nd carbon atom in the 1st pyrrole ring. This is the only difference between chlorophyll A and chlorophyll D. Okay. Chlorophyll molecule is formed from protochlorophyll in presence of light. The protochlorophyll lacks 2 hydrogen atoms, one each at 7 and 8 carbon of 4th pyrrole ring. So protochlorophyll in presence of light and hydrogen give rise to matured chlorophyll, and this reaction is catalyzed by enzyme called protochlorophyll oxidoreductase. So next pigment, which is an accessory pigment called carotenoid. Carotenoid is yellow, brown, orange or red pigment found in photosynthetic cell. The carotenoid also provide color to fruits, flowers because they are present in the chromoplast. These different colors of fruit will attract the insect, and pollination helps in pollination. The most important function of carotenoids seem to be protecting the plant from free radical formed by ultraviolet rays or other stress condition. Hence, carotenoid is also called non-enzymatic antioxidant. There are two types of carotenoids, carotenes and xanthophylls. Coming to carotenes, carotenes are yellow or orange pigment. They are open chain conjugated double bond system ending on both sides by ionic ring. The structure is given here. They are open chain system end at the both sides by a structure called ionic ring. So their chemical formula is C40H56. Different carotenes different only in the arrangement of molecule in space. That is, the carotenes are stereoisomers of each other. The type of carotenes, different carotenes are stereoisomers. In lycopene, which is a chief pigment in tomato fruit, the ring at the both end of its structure are open, not closed. It's the open ring are seen in lycopene. Carotenoid absorb light maximally between 460 nanometer and 550 nanometer and appear red, orange or yellow to us. So these are the different structure of different carotene: alpha carotene, beta carotene, lutein, zeaxanthin, beta cryptoxanthin, lycopene, etc. Different carotene only differ by the arrangement of the molecule. They are stereoisomers. Coming to xanthophyll, or it is also called carotenol. They are similar to carotenes. Only difference is that they are having two oxygen atoms in the form of hydroxyl, carbonyl or carboxyl group attached to the ionic ring. General formula of xanthophylline is C40H56O2. In some type of xanthophylline such as violoxanthin, due to epoxidation of the ring, the general formula will become C40H56 and O4. These are the structure of several carotenes. That is violoxanthin, lutein, zeaxanthin. The structure is similar to that of carotenes. So, apart from their role of absorption of light and transferring to chlorophyll A, these carotenoids also play very important role in preventing photodynamic damage within the photosynthetic. So the third and last type of pigment called phycobillins. Phycobillins are red and blue pigment. They consist of open conjugated system of 4 pyrrole ring which lack magnesium and the phytol chain. The water soluble pigment. They are water soluble and therefore they are seen in cytoplasm and stroma of the chloroplast. Generally, these phycobillins are observed in cyanobacteria and rhodophyta. So there are two different types of phycobillins. These are the general structure of phycobillin, that is phycoerythrobilin and phycocyanobillin. Two types of phycobillins are seen, phycocyanin and phycoerythrin. So phycocyanin is majorly found in cyanobacteria and phycoerythrin is present in red algae. This phycocyanin and phycoerythrin are seen both in red algae and cyanobacteria, but the amount of phycocyanin is more in cyanobacteria comparing to red algae. So these are the photosynthetic pigment. These are the principal pigment of photosynthesis is chlorophyll A, and in the case of bacteria, bacterial chlorophyll A. And accessory pigment are chlorophyll B, C, D, E, F, etc. and carotenoids and phycobillins. So carotenoids are of two types, carotene and xanthophyll, and phycobillins are of two types, phycoerythrin, phycocyanin. And another type of phycobillin called allophycocyanin is also present. So coming to the location of photosynthetic pigment in the chloroplast. Generally, this photosynthetic pigment is located in the grana portion of the chloroplast. Their hydrophilic head of the chlorophyll molecule remain embedded in the protein layer, while the lipophilic tail which is seen in the lipid layer. So, as we all know, the membrane of the thylakoid is lipoproteinaceous in nature. That is, lipid and protein are present in the membrane. So, the head region, in the chlorophyll pigment, hydrophilic head region of the chlorophyll molecule remain embedded in the protein layer, which is also hydrophilic, while the lipophilic region is embedded in the lipid region. So coming to absorption and utilization of light energy by photosynthetic pigments. As we all know, different pigments absorb light of different wavelength. Okay, so the light energy that we get is from sun's energy. Chief source of light energy of photosynthesis is sun. All the incident light falling in green part of the plant is not absorbed and utilized by pigment. Some of the incident light is reflected. Some of them is transmitted through them, while only small portion is absorbed by the pigment. Photosynthetic pigment absorb light. This is the light spectra. So the photosynthetic pigment absorb light between the wavelength 400 to 700 nanometer. So such radiation is called photosynthetically active radiation, or PAR. So the radiation which is useful in photosynthesis is called photosynthetically active radiation. So only 1% of the total solar energy reserved by the earth is absorbed by pigment and utilized for photosynthesis. There is a very weak absorption by pigment in green part of the spectrum. Hence, the chloroplasts appear green in plants. Coming to absorption spectrum, a plot showing absorption of light of different wavelength of a pigment is called its absorption spectrum. Both chlorophyll A and B show maximum absorption in the region of violet and red of the absorption spectra of chlorophyll and carotenoids are shown in this picture. So chlorophyll molecule absorb the from the region of violet blue region and also from the red region. So the absorption band seen in the violet blue region is called the Soret band. Carotenoid absorb light energy in blue, blue-green and green part of the spectrum. Chlorophyll B is and chlorophyll A is having almost similar type of the absorption spectra. So these are the absorption spectra of chlorophyll and carotenoid. Absorption of chlorophyll A show peak at 680 nanometer and that of chlorophyll B at 450 nanometer. Carotenoid show absorption at 478 nanometer and xanthophyll at 440 and 400 nanometer respectively. So next coming another type of spectrum. What is that? Action spectrum. The plot showing relative effectiveness of different wavelength of light in photosynthesis is called action spectrum. So we have given the raw material called light to the reaction center, and reaction center should give the productivity. So productivity is the action spectrum. Relative effectiveness of different wavelength of light in photosynthesis is called the action spectrum. Absorption spectrum is given by all pigment, but the action spectrum is only given by chlorophyll A. Why? Because chlorophyll A is only having reaction center. All other pigment are accessory pigment. So there is no effect or there is no byproduct or there is nothing in another pigment, but only chlorophyll A is having some action or they are the reaction center and they are doing the photosynthesis. Hence the absorption spectrum is given by all the molecule, but action spectrum only by chlorophyll A molecule. The action spectrum of the chlorophyll A molecule is shown in this figure. Okay, so there is the similarity between absorption spectrum and action spectrum of the chlorophyll A. The peak showing that is the more absorbed region, the light where the light is more absorbed, in that region itself action, that is reaction is more. The peak of action spectrum have more height and value are much lower, narrower as comparing to absorption spectrum. Okay. So the action spectrum of chlorophyll A is much more efficient than its absorption spectrum. This is an indication of the fact that chlorophyll A perform more photosynthesis than it actually absorb light. So chlorophyll A only absorb a limited amount of light. All other light which is used for photosynthesis is transferred from accessory pigment to the chlorophyll A molecule. Hence, it is the indication, the fact is that the chlorophyll A, the performance of chlorophyll A is more than actual is what it is absorbing. Almost all pigment except chlorophyll A are called accessory pigment or antennae pigment. So, almost 95 to 99% of the light absorbed by the accessory pigment is transferred to the chlorophyll A molecule by resonance, also called as Foster transfer. Chlorophyll A molecule also absorb light directly. So, what I said earlier, that only a special form of chlorophyll A called P6IT and P700 can take part in primary photochemical reaction in the photosynthesis. All other chlorophyll A molecule also act as the accessory pigment or antennae molecule which absorb light and transfer it into the reaction center, which is called P680 and P700. That constitute the reaction center in PS2 and PS1 respectively. So next question which comes in mind is that how this accessory pigment transfer light into the chlorophyll A molecule or reaction center. Since all they are absorbing same sunlight but of different wavelength. As the accessory pigment absorb the light of lesser wavelength and emit the light of higher wavelength, and this process is by photoluminescence. So it is the phenomena of re-radiation of absorbed energy, and photoluminescence are of two types, fluorescence and phosphorescence. So fluorescence and phosphorescence. Fluorescence and phosphorescence are the phenomena which takes place in the reaction center of the photosynthetic region. We all know the energy that the reaction center obtained is from the sunlight. So when the sun energy hits the molecule or atom which is in a ground state, that is in a normal state, they get excited. When the high energy is given to that molecule, they will become excited and reach a stage called second singlet state, which is a highly unstable state. In the second singlet state, they are unstable and it loses some amount of energy and reaches a state called first singlet state. From first singlet state, by two ways, this molecule can reach the ground level. First one is, it can lose the energy, that a high amount of energy which is seen in the excited molecule, in the form of heat energy. That is, by avoiding some energy in the form of heat energy, and they can reach into ground level. Or in second way, they can radiate some amount of energy in the form of radiation energy, that is light energy, which is higher wavelength of that of the incident light. That the second way of losing the energy in the form of radiation energy or light energy is called fluorescence. So fluorescent light is having higher wavelength that of the incident light, and the fluorescence occur only during the period when they are exposed to the incident light. So this is called fluorescence. So I hope all understood what is fluorescence. Second one is phosphorescence. The excited molecule or atom may lose the electric excitation energy by internal conversation and come into another excited state called excited triplet state. From the excited triplet state, they can reach the ground level, by two methods, same as that of fluorescence, by emitting some amount of energy in the form of heat or by emitting radiation energy. That radiation energy emitted in the triplet state is called the phosphorescence. During phosphorescence, the emitted light will be higher wavelength of incident light and also higher wavelength of the fluorescent light. Okay, that is the phosphorescence. So all the photosynthetic pigments show the property called fluorescence. What is the significance of fluorescence in photosynthesis? The significance is that the phenomena of fluorescence has greatly helped in understanding the transfer of light in between photosynthetic pigment by resonance. That is, some photosynthetic pigment absorb the light of shorter wavelength and they emit the light of longer wavelength. For example, a molecule which absorbs the light in the 400 nanometer region may emit the light which is 500 or 600 nanometer of light. In such cases, the fluorescent, and it's not difficult to understand that fluorescent spectrum of some pigment will overlap with the absorption spectrum of other pigment, such that the accessory pigment having a fluorescent spectrum, that is, they emit the light of a particular wavelength which comes under the absorption region of the another molecule or absorption spectrum of the core complex. So this is what happening in the transfer of energy or transfer of light from accessory pigment to the reaction center occur by the phenomena called fluorescence. During fluorescence, they emit the light of higher wavelength which is absorbed by the another molecule which is having the absorption spectrum in that region. So that is the significance of fluorescence in fluorescence in photosynthesis. Okay. So fluorescence is very important in photosynthesis in the light harvesting complex. Since these complexes are compactly packed inside the thylakoid, in the thylakoid membrane, the fluorescent spectra of one molecule is come under the absorption spectra of other. Hence, the light energy can be transferred from the accessory pigment to the reaction center by this process. Okay. So, the light harvesting complex consists of different pigment system which is compactly arranged. So, that it is having two types of pigment system, that is light harvesting complex will have antennae molecule and the reaction center. The light energy obtained from the antennae molecule is passed into the reaction center. The energy is finally transferred into photosynthetic reaction center, and the photochemical reactions are taking place in the reaction center. Okay. So, light harvesting complex is a group of pigment molecules which are associated with each other and help carrying out photosynthesis. Light harvesting complex is made of hundreds of pigment molecules except for only one chlorophyll A molecule forming light harvesting system. Okay. So, light harvesting system is a group of pigment molecules which are associated with each other and help each other to carry out photosynthesis. Light harvesting molecule, or in that the pigment molecule which is present in the light harvesting complex, except only one molecule which is chlorophyll A, all other molecules are antennae pigment. The single chlorophyll A molecule forms the reaction center while other pigment serves as the accessory molecule and make the photosynthesis more effective by absorbing light of different wavelength. So, by this we are coming to the end of this session, and in next class we will discuss about different photosystem, light and dark reaction of photosynthesis. Thank you.